Hi everyone, welcome back to our new unit. This is unit 4.1, and we're going to go over DNA replication, mitosis, and cancer. So there'll be two videos. This is video number 1. We'll do a quick review of the structure of DNA, and then we'll talk about how DNA replicates itself, which is something we haven't mentioned yet. So first, let's review DNA structure, and we're also going to add a little complexity to DNA structure. First of all, remember that DNA is a polymer and it's made of nucleotides. So nucleotides are the monomer of DNA. And a nucleotide consists of 3 parts. We have a sugar, a nitrogen base, and a phosphate group. And you can see my diagram down here, right? We have our phosphate group, we have our 5-carbon sugar, and we have a nitrogen base. Now that nitrogen base can be one of 4 different possibilities. We have adenine, thymine, cytosine, and guanine. And if you remember the complementary base pairing rules, adenine always pairs with thymine, cytosine always pairs with guanine. Now, a new part of DNA structure that we didn't mention before is that DNA we know is a double helix, which means it has 2 strands, but those 2 strands run anti-parallel. What does that mean? Well, first, to understand that, we have to take a look at this 5-carbon sugar. So the 5-carbon sugar, in chemistry, whenever we have carbon rings, carbon skeletons, science numbers the carbons. It helps with naming the compounds and identifying them, etc. So you can see here that every point in my hexagon, my 5-carbon sugar, every point is a carbon. So you can see right here we have number 1, and if we go counterclockwise 2, carbon 3, carbon 4, and carbon 5 right at the top here. So there's our 5-carbon sugar. That's where that comes from. So when we say that 2 strands of DNA run anti-parallel, if we take a look at the top diagram up here, we can see that one side of DNA runs in this 5' to 3' direction, and the other strand runs 3' to 5'. They're running in opposite directions. Now, where does this whole 5', 3' thing come from? Well, it has to do with what number carbon is left freely open to the exposed end. So you can see the 3' section of DNA is so given the 3' end because it's the number 3 carbon that's left exposed. Up at the top here, it's the 5' end because it's the number 5 carbon that's left exposed. On the complementary side, we have the opposite, right? At the top, we have the number 3 carbon that's exposed, and down at the bottom, we have the number 5 carbon exposed. Now, why does this all matter? Well, it has to do with the way that DNA fits neatly together in this nice uniform molecule. It's part of what Watson and Crick had to work out when they were deciding on the structure of DNA and how all the pieces fit together. And we're going to see it's important in how the enzymes replicate during DNA right before cell division. So let's talk about DNA replication here. Why does it have to happen? Well, first of all, remember that DNA contains all of the genetic information for a cell. And when we say all of that genetic information, what we really mean is it contains all of the necessary genes that tell the cell what proteins it should make. And we know now that proteins are the cell's worker molecules. Every chemical reaction, every process that a cell needs to get done, it gets done by using proteins, enzymes, you name it. Where do those proteins, those enzymes, where does all that come from, or the information to make those things come from? It comes from DNA. So before a cell can divide, meaning before we can go from one parent cell to 2 daughter cells, and then 2 daughter cells to 4 daughter cells, and 4 daughter cells to 8 daughter cells, and so on and so forth, and we build a whole complete organism, we have to have DNA replication because those daughter cells are going to need a set of instructions on what to do. They're going to need to know what proteins to make, what enzymes to make, what processes to run, etc. So before any cell can divide, we have to make a copy of those instructions. We have to make a copy of that DNA. Now, DNA replication is a highly regulated process. There are multiple enzymes involved. Each one has their own unique job to perform. It's going to be based on the complementary base pairing rules. So remember A with T, C with G. In fact, it's that complementary base pairing rule that's going to preserve the genetic message between generations. Because what's going to happen in DNA replication is that our original DNA, which you can see over here in green, so the original DNA is going to serve as a template or a blueprint, in other words, to make two new strands from, right? So if we follow here in the diagram, green represents my original DNA. And you can see that the very first thing that we have to do if we want to copy the DNA message, we have to open it up. So there's going to be a special enzyme, we'll talk about it in a second, that opens up the DNA molecule and creates these two template strands. And you can follow the arrow. Here's one template strand, here's the other template strands. So the green is representing the original DNA. We're going to have another enzyme that's going to come in. It's going to read the template strand. So every time it reads an A, it would bring in a T. If it reads a C, it'll bring in G. If it reads G, it brings in C. If it reads A, it brings in T, and so on and so forth. So it's following those complementary base pairing rules. Once that enzyme has finished reading all of the template strands, what we end up with at the end are two identical DNA molecules. One that both of them are each half original DNA and half new DNA. Here, orange is representing our new DNA. This is known as semi-conservative replication. So if we take that apart, semi means half, conservative means to save, to preserve. So semi-conservative replication is where we are saving one half of the original DNA every time we make a new strand. Every time replication takes place, we are saving one half of the original DNA in each new strand. Now, the process of DNA replication is highly accurate. In fact, of like the 3 billion base pairs that are going on in your 6 feet of DNA inside every cell, the the enzymes make maybe one mistake per every 1 billion base pairs. That is super, super accurate. And in fact, the same enzyme that makes those new strands of DNA can actually go back and proofread its work and catch some of those mistakes. So let's look at what actually takes place. We'll take a look at some of these enzymes, give them a name here. So in DNA replication, we're going to start this diagram goes left to right. So here's my chromosome, and my chromosome is a coiled, condensed mass of DNA. So the light blue coloring is the original DNA molecule. Now, I said before that in order for DNA to be copied, we have to first open up that double helix. We can't very well copy the message if the helix is closed up completely. So we have a special enzyme. That enzyme is helicase. Remember ASE, our ending for enzymes? There it is, so helicase. The job of helicase is to unzip DNA. And the space where that unzipping takes place is called the replication fork. Now, there are some other proteins that aren't mentioned in this diagram. They're called single-stranded binding proteins, and they help stabilize the open strands of DNA while replication is taking place. But for you guys, I just want you to remember helicase, enzyme that unzips DNA, and it creates this replication fork. Now, in reality, there are multiple places along the DNA strand where replication is taking place all at once. It simply doesn't start at one end and then work its way down to the very bottom. Okay, there's multiple places where this is going on at the same time, sort of making the whole process more efficient. All right, so now we opened up this DNA and we've got these exposed areas, these exposed bases, right? Well, here's where that whole 5' to 3' thing comes into play. DNA polymerase, another enzyme, there's our ASE. DNA polymerase, if we broke down what this means, literally means polymerase. Anytime you see an enzyme that says polymerase, it means that it's an enzyme that's involved in building something big. It's an enzyme that's involved in building a polymer. So in this case, what's the polymer we're building? We're building DNA. So DNA polymerase can only add nucleotides in a 5' to 3' direction. That means that the 3' to 5' original DNA becomes what is called the leading strand. And the leading strand is a strand of DNA, it's the strand of newly synthesized DNA that is built continuously. So you can see this red arrow right here. The leading strand here, DNA polymerase, as soon as a nucleotide is opened, it can add the complementary one. And it builds this new DNA molecule in a continuous fashion towards the replication fork. So as this continues to unzip, the leading strand just continues to be synthesized. Down on the bottom strand, though, we have a problem. Down here, DNA polymerase is forced to wait for a free 5' end, which is up here. So it has to wait for the molecule to unzip before it has another free 5' end. So the lagging strand is called the lagging strand because it's actually built in the opposite direction. Okay? It's built being facing away from the replication fork, and it's built in short fragments called Okazaki fragments. And Okazaki, it's named after the scientist who first witnessed this process and documented the process. But the leading strand is built in short pieces. So like here, we'd have this short little piece built, and that would be one fragment. And then the DNA polymerase would start up here and build another fragment. And then as more of the DNA unzips, it would build another fragment. And as more unzips, it would build another fragment. And all of these Okazaki fragments, when everything is all said and done, they get sealed up back together. There's another enzyme called ligase that would come in and seal all of the fragments together so that we have a nice uniform new strand of DNA. But so again, DNA polymerase is the molecule that's actually adding complementary nucleotides to these template strands and building those new DNA molecules. We have a leading strand that's built in the direction towards the replication fork. It's built continuously. And we have a lagging strand that's built discontinuously. It's built in fragments called Okazaki fragments. But at the end, we have two, if I go back to this picture here, in the end, we have two identical DNA molecules. Nothing has changed about the message. The cell didn't change up the base pairs, didn't change up the order or arrangement. Two genetically identical DNA molecules that are going to be half original DNA and half new DNA.